email address. See what happened.
It says that it's being live streamed.
Okay, that was weird. I got an error I've never gotten before, but I think we're live on YouTube, so the recording is good to go. It's all fine. Maybe we should start because we have 2 speakers right today. Okay. So welcome everyone for another session of Cell Migration Seminar. Today, our first speaker is going to be Dr. Suraj Shankar, who is currently a junior fellow at Harvard Society of Fellows. He obtained his PhD in physics in Syracuse University, during which he also was a graduate fellow and a visiting affiliate at Kavli Institute of Theoretical Physics in Santa Barbara. His research interests are broadly in soft matter and biological physics. His current work is focused on active matter control theory, extreme mechanics, and morphogenesis. Suraj, have to tell me what is extreme mechanics because I don't know that. Today, he is going to talk about viscoelastic manipulation of collective bacterial swimming. So over to you, Suraj, and thank you so much for accepting our invitation.
Well, I mean, thank you for asking me to be here, and for everyone to in the Zoom call and on YouTube as well. And thank you for organizing this series. I mean, if not on oh wait, what happened? I do try to follow as many of the previous talks that I can, and so it's been a great series throughout COVID. Very, very helpful for the entire community. So thank you again. And so as Ankita mentioned, I will be telling you about a story that we recently finished on bacterial swimming, collective bacterial swimming in viscoelastic suspensions, and how viscoelasticity and the rheology of the fluid in which the bacteria move can actually have interesting dynamical consequences on self-organization of bacterial collectives. And so just to start with a bit of motivation, and this is a sort of generally, we all know, and I think we all appreciate in various different settings, that cells in both eukaryotes and prokaryotes, and they navigate extremely complex environments, both spatially and temporally. And I've just given a few examples over here, sort of in the context, particularly of bacteria. For instance, Helicobacter pylori, which is it's a bacterium that's a pathogen. It's a pathogenic bacterium that lives and resides in the gut and stomach mucosal lining, and it is, of course, responsible for extreme gastrointestinal problems such as ulcers. And over here you see these tiny little Helicobacteria pylori sort of swimming in this in mucin, and they do so by actually digesting and destroying the local polymeric matrix, allowing them to swim through it. Of course, a different example, not bacteria here, but sperm also navigate extremely rheologically complex fluids. In this case, bovine sperm in the cervical mucus, swimming through again a mucosal lining, and in this work people have shown that now viscoelasticity of the mucus matrix enhances, first of all allows sperm to sort of cluster and form these small clustered patches that can actually enhance collective swimming and migration. And of course, one can imagine that this is a very important property and very important phenomena of deep relevance to problems in fertility. In slightly different contexts, in more spatial and more solid complex environments, of course, bacteria inhabit soil, biofilms that they create by themselves by secreting extracellular matrix. But also in this case, a synthetic porous medium of hydrogels from Sujit Datta's lab, but also bacteria, swim in mucus linings and various other an extracellular matrix in the gut microbiome as well, and in various different settings. So both natural, in within organisms, outside as well, and of course, even various other eukaryotic cells move and navigate through ECM, and you might hear some of this I believe, in the next talk in the context of both cancer invasion and mesenchymal cells. And so this is a very common feature of migration, both individual and collective, in complex and responsive environments. And so while a lot of work and focus has been in looking at individual motility traits and if you want, locomotory gaits of individual cells and how they respond to their environment, I want to think more in terms of the collective. So ask, in the context of bacteria, for instance, what happens when you have many bacteria swimming around? So what are the collective self-organization properties? And how does a responsive environment further affect this self-organization? In particular, when the environment also is now not just static, but can dynamically change itself and adapt and respond to the dynamics and swimming motion of the bacteria as well. And so to just largely recapitulate what we know and understand about bacterial self-organization, I will just be rather brief and quick over here. So, of course, single bacteria, where a cartoon of E. coli does this very characteristic swimming style, which is run and tumble. So it moves in long straight runs and then tumbles in some stochastic fashion by changing, by unbundling its flagella and then turning around in some random orientation. That's a single bacterium. Now, when you have collections of these, one thing that we do know from various different studies, both theoretical and experimental in the past several years, is that collections of swimming bacterium, they generate fluid flow because the flagella are beating and then they push fluid around them. And so when you have large number of these swimming microorganisms together, the fluid flows that they self and actively generate cause reorientation of the bacteria themselves and destabilize local clusters of ordered swimming cells. And this leads to phenomena that is now fairly ubiquitous in dense suspensions of active swimmers of various kinds, and has been called bacterial or mesoscale turbulence. You have, in this case with B. subtilis, in this example video, you have this large scale sort of coherent swirling motion of a dense collection of cells over here that is reminiscent of turbulence in regular fluids, but is actually quite distinct. Particularly, was that a question? Okay. So you have this sort of large-scale, spatiotemporally chaotic swirling flows that are actively generated by the bacteria as they're swimming. And so one immediate question is, how does one sort of rectify this almost chaotic swirling behavior into something that's more coherent, more ordered? And one way to do it is to confine the whole suspension. So if you confine bacteria into a small drop on the scale of 10 to about several tens of microns, up to about 100 microns, you can stabilize collective motion of the swimming bacteria. And in this case, simply by virtue of the geometry of the confinement, the bacteria are swimming around in a circle. So it's like a vortex. It's a rotary vortex. But then the moment you make this confinement weaker and weaker, you have a large drop, then eventually this ordered swimming devolves and dissolves into something more turbulent and chaotic, like in these unconfined suspensions. And so if you want to have sort of collective ordered migration on large scales of bacteria, confinement alone is not quite enough. And then an immediate question, a natural question, sort of along the lines of what I initially spoke in the motivation, is that what happens now when the surrounding fluid in which the bacteria are swimming is not just a simple solution like water, but has some intrinsic dynamics of itself. And so in particular, if you have a fluid which is viscoelastic and can respond, can store and respond in terms of mechanical stresses, what does that do to this collective organization of dense suspensions of bacteria, and how and when does it stabilize, destabilize various kinds of ordered migration of bacterial collectives? So that's
I have a very
Claire has a
I have a
question.
very quick question to that I need to answer to understand things.
Absolutely.
Do the flagella of bacteria entangle with each other at all?
Entangle with, that's a good question. The
Or do
simple
they
answer
stay like that cartoon you drew?
I actually don't quite know. So I guess eventually, if you have a very, very dense suspension, yes, you could potentially have entanglement of the flagella. My guess is, at least at the concentrations that are typically used in the experiments as I'm showing here, and in the ones that I will show you in the next few slides, you probably don't have direct contact between the flagella. And you probably don't have direct contact between the bacteria themselves. And so entanglement between flagella is probably not quite happening at these stages, but it's potentially
But
relevant.
you didn't consider it, is what you're saying, basically.
Yeah, we don't consider it, and
Okay.
but you might need to go to much higher densities and to have an actual entanglement of flagella. Yeah. Thank you. But no, thank you. Thanks for the question. And yeah, feel free to interrupt and ask questions along the way. Yeah. Okay. So this is what I want to address in the rest of this talk. And so along with our collaborators in the Chinese University of Hong Kong, so Yilin Wu and Song Liu, who was then a grad student with Yilin, and now is a postdoc at IBS in Korea. So they did these, fantastic, remarkable experiments with very dense suspensions of E. coli in confined to a sort of thin puddle, if you want. It's a large drop. The diameter of this drop is on millimeter scale, so this is gigantic. It's about 1.5 millimeters. The thickness of the drop, or its height, is basically about 20 to 30 microns. So it's extremely thin. It's an extremely almost thin layer of this bacterial suspension. And if you just have a simple bacterial suspension of that kind, then you actually just get the kind of bacterial turbulence that I showed you before, large-scale swirling motion. But now if you add small amounts of extremely high molecular weight, an extremely high molecular weight polymer, in this case E. coli DNA itself, which has a molecular weight in the giga-Dalton regime, so it's extremely high, and small amounts of this polymer, when added to the suspension, can actually remarkably stabilize large-scale ordered and coherent laminar flow. And in this case, it's collective flow. If you can see, all of the bacteria collectively swimming essentially in a giant laminar vortex, and this is on the millimeter scale. So this is on scales much larger than people have ever been able to stabilize bacterial flows previously. And this situation is extremely stable, and so in a sense, by additions of small amounts of viscoelastic, by having small amounts of viscoelasticity, you can sort of calm turbulence. And this is, in a sense, reminiscent of an effect that is well known in inertial flows, wherein adding polymers in inertially turbulent fluids leads to what's called drag reduction in polymer-induced calming of turbulent flows. And so here you have essentially an active variant or a bacterial variant of something similar, which is where by adding small amounts of viscoelasticity, you actually stabilize coherent swimming. Now, if you increase the amount of polymer concentration, you get something even more striking, which is that this whole gigantic millimeter scale vortex actually starts switching periodically. So you get these periodic oscillations of on the scale of a second to tens of second oscillation frequencies. And here you're seeing essentially in phase contrast microscopy, this vortex of bacteria collectively swimming is sort of switching direction. And here you can do PIV and measure the velocity field, and which makes it sort of very clear that the orientation of the flow keeps periodically reversing. And so how can we try to understand this? How do we, just by tuning the concentration of the amount of added polymer to the suspension, the collective self-organization and swimming of these dense bacterial suspension gets sort of rectified into a large-scale coherent motion on the scale of millimeters. And then eventually, when the viscoelasticity is strong enough, you actually get periodic reversals of flow. And so how can we start to understand this? So for that, we built a fairly simple, well, it looks complicated, but the actual physical ingredients are quite simple. We built a model, a continuum hydrodynamic model, which to describe what's happening, and it's largely phenomenological. And these two slides, this and the next slide, are going to be a bit mathematical, so bear with me. But hopefully, the underlying physical mechanisms are hopefully more intuitive and clear. So to describe this system, we use basically a couple of continuum fields, if you wish. One is to essentially describe the average orientation of local collections of bacteria that are swimming. And so you can think of that being a vector or a polar, it's like an order parameter, it's like a polarization, it's the average direction in which all the bacteria point. That has some dynamics in which bacteria want to line up and swim together. And also they get reoriented by flows in the surrounding, which is captured by shear and vorticity in the fluid. The whole suspension itself is all of the swimming dynamics happens at low Reynolds number. So we work in the Stokes regime without inertia. And so force balance just dictates that viscous forces are balanced along with pressure in the fluid for incompressibility, friction with the substrate because there's an extremely thin drop. And so those are all passive, essentially mechanisms of drag. Where are the active mechanisms which encode the ability of the bacteria to swim? One is its own self-propulsion speed, which is that bacteria want to swim at a given speed along the direction in which they're heading. So that's this V naught. And in addition, the fact that these bacteria are swimming in a fluid means they exert stress in the fluid, because E. coli in particular are pushers, so they exert a force dipole on the scale of the length of the bacterium, and the average density of these force dipoles leads to what's being called an active stress, and that's what is shown here. So these are, if you want to think of this fluid is suffused with a bunch of swimming force dipoles, and that's what this model essentially captures.
I have another question.
Yes.
Are they still trying to tumble at some frequency, or have you negated tumbling?
Yes, so tumbling is sort of captured, is basically a reorientation of the direction in which they're moving. And so that's captured by some decay rate, which I've not quite explicitly written, but it's included in the model. Yes, thank you. So this is just, in the absence of the polymer, in the absence of viscoelasticity, there's a very, perhaps complicated looking, but it's actually, it just includes some very basic minimal ingredients of what the bacteria actually are doing. Now, when you add the polymer, which is you add some viscoelasticity to the medium, then in addition to the orientation in which the bacteria are swimming and the background fluid flow, we have to now take into account the dynamics of the polymer itself. And a simple way to account for the polymer is the stress that the polymer can experience. And so there's an elastic stress of the polymer, which obeys essentially a version of the Maxwell viscoelastic model. There's a characteristic relaxation time, which is the Maxwell time, on which it's a time scale on which the polymer relaxes internal stresses. But in addition, we phenomenologically include a coupling between the bacteria and the polymer, which is essentially saying that on a time scale, tau r, the bacteria want to realign to the stresses in the polymer. So now you have an environment, which is the polymer, having its own dynamics on a given time scale, and the bacteria can respond to this coupling to the environment via this additional stress feedback, if you wish. And so we can take this sort of continuum model and just put it, run a numerical simulation of it. And so what we can tune is now the relaxation time of the polymer. So for small amounts of viscoelasticity, the relaxation time is very small. And what you find is that you can actually stabilize the swirling, turbulent, and unstable flow of the bacteria into this ordered vortex, exactly like we see in the experiments. This is the polarization. So the bacteria are all pointing along these arrows, and here's the flow velocity, which you also see is a vortex. And this is just a plot of the azimuthal flow velocity as a function of radius. And so this is consistent with what we see in the experiments. And now if you tune, if you increase the amount of viscoelasticity in the model, so you increase the Maxwell relaxation time, then remarkably what you get is, oh, no, the next slide, sorry. What you get is now spontaneous oscillations. Here you can see that the flow velocity generated by the swimming bacteria actually reverses, very much like in the experiment. And let me run that again. And so you get this oscillating vortex. And the nice thing you can see, this is a time trace of the average azimuthal flow. It's periodically oscillating, and it is correlated with distortions of the orientation in which the bacteria are swimming. And so that's what's shown over here. Splay is essentially, splayed out distortions. So although the flow in the vortex is flipping sign, it's switching direction, the bacteria don't reverse 180 degrees, but rather it's only the average orientation in which they're swimming gets locally distorted. And so you get this, as you can see here, the sort of breathing deformations of or distortions of local bacterial orientation, which are coupled with reversals of vorticial motion. And so this is also consistent with the experimental observations. Now, of course, we did more serious quantification of and comparisons, which I'm not showing you here. But how can we sort of distill the actual mechanism of both stabilization of large-scale coherent motion and the origin of the oscillations? So to do that, I will just sort of sketch a very simple conceptual picture. So of course, you have two transitions, and so the easiest way to get to transition is by balancing three timescales, right? So there are three relevant timescales in this problem. One is the timescale of the environment, of the polymer itself, which is the viscoelastic Maxwell relaxation time, the time on which the polymer relaxes elastic stresses. There is an intrinsic timescale for the bacteria, which is an active shearing time. It's the time scale over which the bacteria pump in energy and generate flows in the system. And that depends on the concentration of how many bacteria you have. And finally, you have now a coupling between the bacteria and the polymer, which is a sort of a reorientation or an alignment time. It's the time scale on which the bacteria recognize that the polymer is deformed and reorient in this background stress field. And so for small amounts of viscoelasticity, when the viscoelastic relaxation time balances the reorientation time, in that case, all actively generated flows by the swimming bacterium distort the polymer, but are immediately accommodated on the time scale. Because these two time scales match, they're immediately accommodated by reorientations of the bacterium. And so you basically allow, so the polymer, by doing so, essentially allows for stabilization of these large-scale chaotic flows, simply because it essentially it enhances the drag that the bacteria experience, and thereby calming the turbulent flows. If you have higher viscoelasticity, so the maximum relaxation time is now stronger, the polymer has the capacity to now store for a significant period of time, store elastic distortions, and then provide it back to the bacteria at a later time. And so you have a delayed feedback with a negative feedback, which essentially, so you have this as the bacteria generate flows which distort the polymer stores this elastic distortion for a time scale tau, and then feeds it back to the bacteria as they reorient. And because of this delay now on longer time scales, you eventually then get oscillations. And so this, you can show, at least within a simple linear stability calculation, you can show that this leads to spontaneous oscillations of the active flow. And you can also obtain a characteristic frequency, which is essentially the geometric mean of the active time scale and the reorientation time. And so you can use this simple conceptual picture to sort of write out or draw out a phase diagram, if you wish, with the axis being DNA, so that's controlling how viscoelastic the medium is, and the other being the density of bacteria, so how active or how dense the bacterial suspension is. And qualitatively, we can capture the rough trends of the phase boundaries of transitions from the laminar vortex into the oscillating vortex, which is like this blue line over here, and the other one is from turbulence to laminar vortex, which is this gray bar. And we can also capture the dependence of these lines and the dependence of the frequency on the DNA concentration. In particular, the longer the Maxwell relaxation time is, the longer the oscillation. So viscoelasticity, which induces this delayed feedback, also controls quite directly the oscillation frequency. But there are some other aspects that we don't quite capture quantitatively, and so that is still work under progress. And so with that, I would like to conclude. Thank my collaborators, Christina Marchetti, Yelin Wu, and Song Liu. And if you're interested in further details, feel free to either email me or have a look at the paper that we published last year. And thank you, and I will take further questions now.
Thank you so much, Shankar. We have a couple of questions. So the first question is from Rhoda Hawkins, asking, is your viscoelastic stress equivalent to Cruz et al.'s model?
Say it again, say the last bit again.
Yeah, so is asking, is your viscoelastic stress equivalent to Cruz et al.'s model?
I'm not so sure which Cruz et al.'s model they're referring to. I know that Carsten Cruz and company have a number of different works. They've done a number of different project works on viscoelastic active systems, and in some contexts, in some regimes, yes, it is reminiscent or equivalent to the kinds of things that they have done.
Yeah, I mean, we can ask if Rhoda wants to unmute,
Yes.
and ask it. Yeah.
I mean, you've basically answered my question. I mean, obviously, Carsten Cruz et al.'s models are often to do with the cytoskeleton rather than bacteria. But in their original cytoskeleton models, they included viscoelasticity. And I just wanted to know whether the way that you included viscoelasticity in your model was equivalent to what they did. I think it was, but I couldn't do the exact comparison in my head, but I just wanted to check.
Absolutely. Yes. So you're absolutely right. So, very similar in spirit kinds of models people have used, and one can use even in the context of cytoskeletal dynamics, but also, sort of mesenchymal or epithelial cell migration in other ECM contexts. So, different scales, the same kinds of couplings and models are very prevalent, and so potentially similar kinds of phenomena are also, viable in other contexts as well. So absolutely, thank you.
Great, thanks. All right, we're running a bit short on time, but we have a multi-part question from Claire Waterman. She's asking, does the splaying and flipping direction have anything to do with tumbling? I was wondering that as well. And does the viscoelasticity affect the tumbling frequency? Does the flipping frequency have anything to do with the tumbling frequency, right? So it's a random process, but there should be average. It should be an average.
Yeah. So I don't think the flipping of the vortex has much to do with the tumbling frequency, because they're very different, at least in order of magnitude, very different timescales. And but the question of whether the actual tumbling frequency of the bacteria changes when you add the polymer, that's a very good question. And I don't think Gillen and Song have any data on that. But that's something that one should actually check to see if we can measure. And asking about whether the splaying of the orientation has anything to do with tumbling, also a great question. And this is something that we've been asking, we've been trying to do. COVID was put a spanner into some of those ideas. So one thing to do would be to fluorescently label the flagella, for instance, and then actually measure not just the flow, but then the orientation of the bacteria themselves, and then you can.
There must be mutants that don't tumble.
Yes,
sure.
The non-tumbling variants are there, and you can play with that. Absolutely. So these are some of the kinds of things that I think they have restarted doing now. But yeah, great question, and something that we will probably continue thinking about and working on. But I don't have an easy answer now.
Thank you so much. We will take another question from Thomas Schmidt. You said that the effect solely occurs in a thin sheet that is like quasi 2D. How does this come in the back of your model? Sorry, it's my.
Oh, in the model, it essentially enters as a friction, because if you're in contact, the whole thing sits on agar, and so our model is itself two-dimensional, and the fact that it's sitting on a substrate means you can lose momentum into the sink, which is agar. That's all. So it's just there's a friction drag that comes because you're two-dimensional and you're in contact with the substrate. But other than that, it doesn't do anything dramatic in the model.
Thank you so much, Suraj. Yeah. Thanks a lot for presenting such wonderful work, and it's sort of very different from what we're used to. So
yeah, I thought so. But thank you for
so much
entertaining me.
I think it's good for those of us who think about crawling animal cells to also think about the other kinds of motility that's out there. So that's really great.
Absolutely. Thank you.
All right. If there are any more questions for Suraj, feel free to put them in the chat or contact him directly. We're a little short on time, so I'm going to move right along to our second speaker. So today we are delighted to have Professor Jacopo Ferruzzi. So he obtained his PhD in biomedical engineering from Yale University. And when he was there, he studied central artery stiffening in aging and multiple cardiovascular conditions. He subsequently moved to Boston University, where he pursued postdoctoral training in cancer mechanobiology. And then in 2020, he became an assistant professor of bioengineering of the University of Texas at Dallas. And he is currently establishing his own laboratory titled the Tissue Mechanics and Remodeling Laboratory. So we're really excited to have him here and be able to hear about some of his recent work. Thanks so much.
Absolutely. Well, thank you very much for the introduction, and thank you for the possibility of sharing my work. Well, first of all, good morning, good afternoon, good evening, everyone. So this work that I'm going to discuss today is part of my postdoctoral work, which took place right before the pandemic, but largely during the pandemic, and was finally published late last year. It will conveniently follow up after last week's wonderful talk by Giorgio Scita and it will deal again with jamming and unjamming transition in cancer invasion. And so I don't think I need to convince this audience that tumors are highly heterogeneous and dynamic tissues. Here you can see, I don't know if I can use my pointer, there you go. Here you can see an image of a tumor explant where tumor cells are labeled in green, whereas the peritumoral collagen is labeled in red. And we know that most of the dynamic take place at the invasive front, where cancer cells, either individually or as clusters, invade through a tumor capsule, which is rich in collagen and stromal cells. So far, less attention has been paid to the tumor core, which is known to be densely packed with cancer cells and low in collagen and stromal cells. However, recent evidence, and particularly this study from a few years ago, show that the tumor core is actually highly dynamic, and then cancer cells display rich collective behaviors, which include coordinated migration, streaming, and vortex-like motions. It is increasingly being recognized that collective migration of cancer cells is the most effective route to spread metastatic disease. So cancer cells can switch their modality of invasion from single cell dissemination to collective invasion to succeed into their metastatic agenda. And the established model to understand conversion of an epithelium from being quiescent to migratory is the epithelial to mesenchymal transition, or EMT. And overall, EMT is thought to result in single cells that detach from the epithelium and migrate away again as single cells. An alternative model that has been recently discovered and still poorly understood is the unjamming transition, or UJT. And this model does not exclude EMT, but it provides a different physical framework to understand cell motility, and particularly collective behaviors. UJT mediates the transformation of an epithelium from solid-like and quiescent to migratory and liquid-like, where this liquid-like phase is characterized by collective migration. At least we know that in one system, the unjamming transition can occur independently from EMT. Now, jamming is a concept from the physics of granular materials. So I'm just going to give you a brief intro to jamming. And in the context of biology, it's been implicated with epithelial remodeling. Here you're seeing a movie of human bronchial epithelial cells provided by Jennifer. These cells cultured on air-liquid interface. And as you can see in this movie, not much is happening. The monolayer is jammed or solid-like. Cells are not motile, and their motions are caged by their surrounding neighbors. Conversely, the unjamming transition, or an unjammed phase or liquid-like phase, can be actually triggered by a variety of stimuli. Here you're seeing a physical stimulus such as compression, but also radiation induce this transition. And as we learned last week, for instance, even expression of an endocytotic marker such as RAB5 will induce unjamming. And in the unjammed phase, the collective, while still being confluent, the cell collective becomes highly motile and adjacent neighbors swap positions. Some of this early work on epithelial monolayers was explained by using the vertex model. In particular, what I'd like to highlight is that one of the main results of the vertex model is that this rigidity transition from solid-like to a liquid-like phase is mediated by a critical value of cell shape, as measured by the aspect ratio, the ratio between length and width. And so as the overall collective, the cell shape of the overall collective overcomes this critical number of 1.18, the cell collective will turn from solid-like to liquid-like. Now, but how does this all apply to cancer? We found recently, the Fredberg lab recently reported that more aggressive cell lines show significant structural signatures of unjamming, while more benign cancer cells are more jammed. But however, this was all valid on 2D. Whereas in vivo, we know that invasive phenotypes can differ dramatically depending not only on the properties of the cells, as shown here, but also depending on the properties of the microenvironment. And this led us to formulate the specific questions of this study, which is, are phase transition implicated in cancer invasion? And if so, what is the role played by the tumor ECM? Now, at the same time as we were formulating these questions and conducting experiments, another study also came out, and I'd like to put their findings also in context so that we can move on to our own results. And so, first of all, I'm going to go back, providing a brief history of the jamming phase diagram. Basically, based on the early work on epithelial monolayer, it was proposed that cellular collectives can behave as soft glassy materials, which can transition from a liquid-like to solid-like states without experiencing the structural ordering that we see in materials at thermodynamic equilibrium. So therefore, the jamming transition is a non-equilibrium phase transition. And for instance, it was postulated that it would depend on several factors such as cell motility, the inverse of cell adhesion, and the inverse of cell density. And so this picture will allow to explain key physical features of migrating cell collectives such as heterogeneity, cooperativity, and criticality. Fast forwarding a few years, a few studies from the group of Peter Friedel have shown that radically different migratory modes can be achieved by modulating the strength of cell-cell adhesions and by varying the degree of extracellular confinement. And so in particular, in a study that came out in 2020, Elena and colleagues proposed or sketched a jamming or hypothetical jamming phase diagram, which is a function of the inverse of cell density and the inverse of cell-cell adhesion. So basically, according to this phase diagram, a solid, a jammed epithelium can unjam and invade progressively by turning into a fluid and then as a gas-like collective. And so therefore, it seems that cancer invasion can be the result of progressive unjamming. And so coming into our work, here we use the tumor spheroid as a model of tumor invasion in 3D. I'm sure that most people are familiar with this model, but I'll briefly review. We form spheroids in low attachment plates for about 48 hours. And once the spheroids are compacted and formed, we transfer them into a matrix of three-dimensional collagen, where we can perform time-lapse microscopy imaging, where we can view and quantify the dynamic behavior of the invasive process. And then at the end of the invasive process, usually about after 48 hours, we can fix, optically clear, and stain our spheroids that can be imaged fully in 3D using multiphoton microscopy, where here you're seeing cells labeled with DAPI in red and the surrounding collagen labeled in green through second harmonic generation signal. I'd like to spend a minute to just describe our pipeline for quantifying or estimating cell shapes. From a multiphoton stack, we can separate the channels, the two channels, the DAPI channel, where we can identify the three-dimensional position of individual cell nuclei, as well as the boundary between the spheroid and the surrounding collagen. And we can combine the information from these two quantification into a bounded Voronoi tessellation, which basically takes the entire tumor mass and divides it into individual cells. Now, conveniently, as we were developing this framework, we were trying to estimate cell shape. A study came out in 2018, a theoretical study, which developed a 3D Voronoi model, which described that phase transition in 3D occur at a critical value of cellular shape, which now is quantified by a normalized surface to volume ratio of 5.41. So as you will see in the rest of this talk, I'll use this value of 5.41 as the critical value that below which the 3D cellular collective is jammed and above which the cellular collective is unjammed.
So just quickly, that tessellation, that's not the actual cell-cell borders. That's just
that is correct. That is
dividing
just
up the space.
That is correct.
So
yes.
Where are you getting shape from if you don't have the cell borders?
Well, so this is, in fact, as I said, this is an estimation. Actually, at the same time, so we validated this into smaller spheroids. Our estimated Voronoi volumes recapitulate the cellular volumes. And also at the same time, a similar study by the group of Joseph Cass, Grosser et al. Showed basically that the quantification by doing accurate segmentation in smaller spheroids basically agree with the Voronoi tessellation. But you're correct that this is not a direct measurement of cell shape. This is an estimation.
Can I also ask a question?
Yes, absolutely.
Is there a minimum size of the cluster that actually you consider a separate phase, or even a single cell, when it exceeds the threshold, would be a phase?
Well, so the whole theory only applies to confluent collective. So whether that is 2D or in 3D, you need to have a confluent collective. In fact, as you can notice, our spheroids show some void areas in the center, and we disregard those. So for instance, we wouldn't be able to apply this area to a single cell, as you mentioned.
Oh, so it's for one measurement for a spheroid?
Yes.
Got it. Thank you.
All right, so going into the results, so in order to investigate whether cell jamming plays a role in 3D invasion of multicellular spheroids, we perform invasion assays using MCF10A epithelial cells. Here you can see two different spheroids cultured in two different collagen density, 2 milligram per ml and 4 milligram per ml, after 48 hours after they were originally embedded. And what you can notice right away is that we do not see signs of invasion in high density collagen, whereas we see signs of collective invasion in low density collagen. And so if we apply our 3D Voronoi tessellation, our analysis, here I'm showing a cross-sectional equatorial cross-section of the same spheroid, where the individual Voronoi tiles are now color-coded by the shape index. And the first thing that you will notice is the largest shape index are estimated at the invasive protrusions. You can actually visualize this data by plotting the shape index versus the radial distance, where 0 represents the center of the spheroid. And here I added the critical line predicted by the 3D Voronoi model, by the theoretical model, as a dashed line. And so what you can notice is that the core of the spheroid tends to show homogeneous shape indices which are close to the jamming transition, whereas it moves further away from this jamming transition towards unjamming at the invasive edge. And now, if we superimpose the quantification on the from spheroids embedded in high collagen density, we see that pretty much the cell shape remains homogeneous throughout the non-invasive spheroid. Now, usually in the theory of jamming, quantification of cell shapes are usually accompanied by quantification of cell dynamics. So here you're seeing one of our phase contrast movies, or DIC movies actually, over 8 hours, where you see that the trajectories estimated by optical flow are shown as red lines. And what you can see is that there is very little motion in the spheroid core, and most of the motion is concentrated right at the invasive edge. And so quantification of RMS speeds now plotted against the spheroid radius, again, agree with our estimates from cell shape, where we have pretty much an homogeneous velocity, and where we see an increase in velocity where the cell shape become more aligned and elongated. And so overall, taken together, these results show that MCF10A spheroid invasion in collagen shows that the spheroid bulk tends to be solid-like, according to this theory, whereas the invasive front is liquid-like, at least in low-density ECM. Now, as we were mentioned, these are just estimation of cell shape, so we wanted to be a little bit sure that we are actually capturing what is actually going on here. And so we teamed up with our colleague at MIT, Ming Guo, who develops microspheroids, what we call microspheroids, that are obtained by culturing single cells, single MCF10A cell in an interpenetrating network of matrigel and alginate. And so we can see that the spheroid at the early stage displays regular shape and rotational motion, whereas at the later stage develops asymmetric shapes and invasive behavior. Now, for sake of brevity, I'm just going to go quickly through these results, but I'm happy to discuss them. In this system, since they are so much smaller than our multicellular spheroid, about 10 to 100 times, we can actually validate the accuracy of our Voronoi estimates of cell volume and associate them with local measurements of cell mechanics via optical tweezers. I'm not going to show this result because they were previously published by Han and colleagues. But basically, if we perform the same analysis on these spheroids, here you're seeing a microspheroid at an early stage on the left-hand side and a microspheroid at the late stage on the right-hand side. If we perform our bounded Voronoi tessellation, we saw similar results, where the shape is pretty much homogeneous in an early stage, whereas we develop spatial heterogeneities at a late stage as this microspheroid becomes invasive. Similar things can be observed by quantifying cellular dynamics by tracking individual cell nuclei. And once again, the results on dynamical measurements agree with our measurements of cell shape, where we see a core that is more consistent with jamming and an invasive edge that is most consistent with unjamming. So once again, even in a different system, in a system with a different dimensionality and a different nature of the extracellular matrix, we see that the spheroid bulk is solid-like, whereas the invasive front is liquid-like. Now, we wanted to account for the possibility of single-cell invasion, as shown by Alina and colleague. And so we performed similar experiment using the post-metastatic cell line MDA-MB31, which shows a more mesenchymal phenotype. As you'll see here, once again, similarly to before, we see 2 milligram per ml collagen on the left-hand side and 4 milligram per ml collagen on the right-hand side. And we see a critical difference in the invasive phenotypes. On the left-hand side, the MDA-MB31 the cells rapidly detach from the main spheroid, adhere to extracellular collagen that is being aligned by the multicellular collective, and invade as single cells, which are reminiscent of evaporating gas particles. On the contrary, on the right-hand side, we see virtually no sign of cell individualization, whereas we see collective protrusions that are radiating from the main spheroid. So now, one thing I'd like to note is that the core of our spheroid, or the MDA-MB31 spheroid, is cell-free, and therefore we had to restrict our analysis only to the cells remaining within the continuous tumor mass, and that are fully surrounded either by neighboring cells or by surrounding collagen. And so once again, we do see that while in low density collagen, the cell shape remains nearly constant, in higher density collagen, now the cell shapes display a radially decreasing trend, which is opposite to what we observed in MCF10A spheroid. So basically, as this multicellular collective is invading, the cell shape become more regular, the volumes become smaller, and this is all consistent with the jamming transition with collective invasion. We found this puzzling at the beginning because this is contrary to what we originally hypothesized. And again, we checked this by looking at cell dynamics. What you can see in our dynamical measurements is that now the tumor core is actually much more active. So if you will, it's more hot. And in low-density collagen, cells will actively migrate within the core and then finally interact with the surrounding collagen, where they will go on and migrate individually following very persistent and directed motions. So this can be quantified only in the low-density case by showing that there are constant velocities, RMS speeds, in the center of the spheroid, and then as the cells detach and come into contact with the surrounding matrix, they actually take off and speed up their migratory behavior. Now, if we only focus our attention within the contiguous tumor mass, so within the black mask that is drawn in both these movies, we can actually compare dynamic behavior between low density and high density collagen. And once again, we see similar features to what we see in our shape measurements, where cell dynamics is fairly constant and homogeneous in low density collagen, whereas we saw a mild but consistently decreasing trend in high density collagen. So overall, this seems to suggest a picture in which the MD-MB31 spheroid bulk is actually more liquid-like with respect to the MCF10A counterpart. And now the invasive front actually behaves differently depending on the extracellular environment. And so we see gas-like behavior in low-density ECM, whereas we see a transition towards a more solid-like behavior in high-density ECM. And to sort of gain a little bit more of an understanding about this process, we looked at things over time. In the interest of time, I'll quickly mention that basically we see that the two spheroids in the two collagen densities start off from similar distribution of cell shape, fairly homogeneous, and over time they develop, while the low density spheroid, they proliferate while still maintaining the same distribution of shape indices. We see in high density collagen this emergence of a radial decreasing trend consistent with jamming. This doesn't seem to be related to cell proliferation, as counts of total cells show no statistical differences between the two collagen densities, whereas the critical fact seems to be cell individualization. With time, in low density collagen, we have a lot of cells that are departing from the main spheroid. And in fact, to understand a little bit how the transition between these different invasion occurs a function of ECM confinement, we use graded collagen concentration, not anymore only 2 concentration, and we quantified the number of single cells that detach from the contiguous spheroid mass. So as you can imagine, at day 0, all the cells are within the primary spheroid, and so there is no single cells. But as we proceed into day 1, and especially into day 2, all the way to day 3, we do see this step-like function, which shows that a lot of cell individualization occurs at low collagen density and much less occurs in high collagen density. And to convince you of that, here I'm showing you some images and 3D renderings of the experimental data that we collected, where you can clearly see where we color-coded the cell collective as blue and single cells as red. And so overall, these findings support the possibility that it exists a critical collagen density somewhere between 2 and 3 milligram per ml, well, where the spheroid periphery transition in almost a switch-like fashion between different modes of invasion. And so we have single cell migration or gas-like migration in low density collagen and collective migration in high density collagen. And this really sedimented the idea that we are witnessing a phase transition. That is, by changing an intensive variable, such as collagen fiber density, we can actually trigger these continuous changes in the state of the system and move from single to collective migration. We characterized the collagen microstructure, and our findings suggested that the collagen fiber density is what is responsible for this phenotype. And we decided to capture the diverse material states and invasion patterns using a computational model that was developed by the lab of Zichan at Dartmouth College. This model shown here is in line with the traditional vertex model, as it includes cell adhesion, motility, cell viscoelasticity, but also it includes now ECM confinement. And so in order to allow cells to migrate individually, as we see in our experiment, we also incorporated an agent-based approach by endowing each cell with its own boundary and allowing it to interact with the ECM, which in a very simplistic way here is modeled as these fixed green posts. And so, motivated by our experimental finding, we decided to vary cell motility on the x-axis here, and the degree of ECM confinement, that is the density of the green posts, on the y-axis, and perform a variety of simulations by varying parametrically these 2 parameters that we identified as meaningful from our experiments. Now, the material state of the spheroid, whether it's solid or liquid or gas-like, was simplistically defined using the 95th percentile of the peripheral cell distance from the centroid. And such simple criterion, along with the 2-state variable, allowed us to draw a phase diagram that captured the solid-like, liquid-like, and gas-like states that we observed experimentally. Now, obviously, this is a very simplistic model, but qualitatively, we can compare the phenotypes observed experimentally with some of the results of the simulations, and we see a pretty good matching and prediction of our experimental results. And again, this model, it's meant to be very simplistic, but it's quite striking that it allows us to reproduce pretty much most of the material states and invasion patterns that we observed experimentally. Now, like any model, it also can predict different states that were not tested experimentally. And for instance, here we can see that it predicts a triple point where solid, liquid, and gas states can coexist. And so to see if this was even realistic in our physical system, what we did is we performed another invasion assay, this time using MCF10A cells, which are characterized by lower cell motility, so they exist on this side of the spectrum, in graded collagen concentration from 1 to 4. And what we saw is that in low collagen density, we can observe cell individualization as opposed to liquid-like collective behavior and all the way to solid-like behavior that are shown by increasing collagen concentration. And so to conclude, the notion of phase separation in tumors is not new. An analogy with the equilibrium thermodynamic system, here we show that we can replace the cell motility with an effective temperature and the confinement pressure and the ACM confinement or collagen density with a confinement pressure. And so of particular importance here are the phase boundary where distinct material phases can coexist. And we find that cancer cell collective can exist along these phase boundaries where they are able to respond to different microenvironmental stimuli by adapting their material state. And so this computational model not only recapitulates some of our experimental observation, but further can suggest that a cancer cell collective can invade the surrounding matrix by following a variety of paths. A solid to liquid, a solid to gas, or a liquid to gas state. And so therefore we suggest that non-equilibrium phase separation based on jamming dynamics can represent a new framework to explore tumor heterogeneity and invasion. So to conclude, we've long known that biological state of a tumor controls invasion. What I showed you here is that the material state actually controls invasion, and the material state is actually dictated by the biological state. In particular, we find that both cell and matrix properties contribute to determine the material state of a tumor and its mode of invasion. It is still unclear how the local material phases impact the biological state. And for instance, an example of some of this deserve further attention. An example of this work was shown last week by George Ushita in their fascinating work. So with that, I'd like to acknowledge the labs that participated to this work, primarily the Freiberg lab and Wendy Kang, who is first co-author with me on this paper. Wendy primarily performed the analysis of cell shape and analysis of cell dynamics, all the way to most of the experimental work happened at Boston University between the Zaman lab and the Robler lab, which helped with multiphoton microscopy. And then ultimately, I'd like to acknowledge the Gua lab at MIT and the Chen lab, and particularly Catalina Paola Spatarello, who performed the computational simulations. None of this would have been possible without funding from NIH. And since I'm probably already beyond time, I'll just leave my contact here. I'll take any question. Thank you for your attention.
Fantastic. Yeah, we do have a number of questions. Do you mind staying a few minutes over to get through as many of these as we can?
Absolutely. I just need to try to see the chat, but yes.
Oh, yes. We'll ask for you. Don't worry.
Okay.
All right. So our first question is from Curtis Scheuer asking, is there a minimum width that the invasive strands must satisfy in order to treat the strand with the same vertex model framework as the larger confluent mass of cells?
No, actually, no. So we are, this is the benefit of using, well, I guess you need to have at least a couple of cells, but that is always the case in our experimental models. This is the convenience of using a bounded Voronoi tessellation, where we need the cells to be surrounded by either neighboring cells or by extracellular collagen. And so this was always the case if you look at our images.
Thank you. Next question is from Abhishek Mukherjee. He's asking, what could be the possible explanation of higher shape index in the early stage microspheroids than the late stage asymmetric ones, and very specifically in the time scale where these two stages overlap?
Well, that is a great question. Our interpretation of these results, it is that at the originally these cells, as you see, they undergo rotational movement similar to the movies that were shown by Giorgio Scita last week, by the way, and basically they exist in a liquid-like state. And then upon further growth over the course of a week to 10 days, what happens is that the cells at the core will experience higher compressive forces and will turn into a solid-like, and so therefore will move down the shape index axis that we drew, whereas the invasive cells now actually can very actively migrate back and forth and show more of a liquid-like phenotype. So we're basically starting from liquid-like and then transition down towards solid-like in the core, and that's what causes that asymmetry.
Great, thanks. Quick reminder to the audience, feel free to do the raise hand function or write question in the chat if you want to ask questions yourselves. Our next question is from Nir Gav. Hi, Nir, asking what is the role of leader cells in the strands?
Well, that is a great question. I don't know that I necessarily have an answer for that. For instance, some of the similar phenotypes were reported, for instance, by the Friedel group, where they see in more mesenchymal-like spheroids that extracellular confinement is sort of enables this collective migration. And there, the leader cells seem to have a role in terms of they proteolytically degrade the matrix and allow the collective to migrate forward. These leader cells then are characterized by specific metabolic features that I know of. And so I believe that there seems to be a recycling of leader cells to further this invasive phenotype. But basically, in this context, we do see that the collectives tend to be slowed down by the process, possibly extracellular matrix proteolysis by leader cells, and so overall show this transition towards a jammed collective.
Great. Next question is from Emma Moore, is asking how plastic is unjamming. If you were to slowly increase the concentration of collagen in the spheroid culture over time, would the single cell transition to a collective migration or more solid-like behavior? I don't know whether that's technically possible or not.
Well, let me see if I understand the question, because technically that's what we try to do, right? We saw this.
But you did it in like separate cultures, right? I think what Emma is asking whether you could, yeah.
Oh, interesting. If you were to slowly increase the concentration in the spheroid culture over time, I am not sure what I would see in that hypothetical case. I would assume that would be true based on our finding and based on what is the current picture, but the truth is that I don't know.
Thank you.
Great. Our next question is from Tina Tootle. Have you looked at how the spheroids are altering the local collagen concentration? And if so, how does this influence the phase transition of the cell migratory behavior?
Well, that's a fantastic question. A question that particularly interests me. I do know, obviously, this crosstalk between the tumor, the surrounding collagen, is a two-way street, and so the spheroid is being directed by the extracellular collagen, but also the opposite happens. And so, if you notice in most of my movies, we do see. Let me see if I can find it. I'm still sharing my screen, right?
Mm-hmm.
For instance, I'll show this. We do find that this is an MCF10A spheroid, where we see that the surrounding collagen is actually densified and show this sort of like locally dense capsule of collagen. So upon spheroid growth, actually the collagen is being impacted, and we see the same phenotype. If you go back, if I go back into the slides, we saw the same phenotype around the MCF10A spheroids. Now, interestingly, the more aggressive, more mesenchymal MDMB31s tend to engage the matrix by aligning the matrix, and so this is a very dynamical process. And so, as of right now, I do not know how we can sort of control this by by isolating the effect of matrix remodeling, but clearly that is something that happens as the tumor either grows or engages the surrounding matrix in tension. I don't know if that addresses the question. Please let me know if I did not really fully answer.
Thank you. Next question is from Claire Waterman. Since you're showing that both jammed and unjammed behaviors can mediate collective invasion, doesn't that seem to show that jamming state is not a relevant parameter for correlating tumor cell behavior to cancer prognosis or survival?
Well, that is an interesting question. As of right now, there seems to be a body of work that suggests that collective behaviors are actually collective invasion. It's actually more functional for metastatic spread, and collective behaviors such as collective invasion have been described quite successfully with jamming and unjamming. The way I see our work, it's actually sort of looking at the tumor mass and basically describing it as a cell collective that lives at the boundary between jamming and unjamming, and depending on microenvironmental condition can actually adapt its migratory behavior according to the microenvironment. And so it can use both phases in order to further invade.
Great. Okay. Our next question, actually, Giorgio Shita, if you'd like to unmute yourself and ask your question, please go ahead.
Hi, Jacopo. Very nice talk. I actually have a quick question. One is related to how much all these phenomena are impacted by the strength of cell adhesion, particularly in light of the fact that you use MCF10A on one end and MDM231 cells on the other, which have a very different
adhesive properties, of course.
Adhesive properties. And the second question, then, on your solid fluid, flocking solid like transition in MDM231, which is very interesting. I love that. I was wondering whether you have analyzed at the beginning of this invasive confined intrusion of cell into the solid flocking state, where they are very confined at the very beginning, before actually they become so, whether you see a more regular type of unjamming-like transition, which eventually becomes a solid-like flocking fluid locomotive.
All right, so let me try to address both questions. So first, your first question is related to the strength of cell adhesions.
Yeah.
So the way we looked at this was intentionally using two cell lines without the opposite side of the spectrum. As you might have noticed, our MDM231 spheroids are quite hollow. They generally do not even form spheroids. Right. So we need to actually form them with a little bit of matrigel that sort of works as a glue that will allow them to stick together. But they really clearly do not like to be together. So if they will find a chance, they will adhere to the collagen matrix and sort of fall apart very rapidly. What was interesting to us was this effect of a very liquid and unstable collective that is highly dynamic, and where cell adhesions are very low, that then can sort of regularize and, as you say, move as a solid flock. And so that was something that was obviously shown also by some of Peter Friedel's work, but we thought that here we had the chance to sort of investigate a little bit deeper how this phase sort of comes into being. And then let me see if I remember your second question. Second question was related to whether we looked at over time as this solid flock develops and whether we see initially signatures of unjamming rather than jamming. Is that right?
Yeah, correct.
We don't seem to see that. Again, our main quantification all the way up here is quantification of cell shape. And so over time, again, we look at this as either maps of cell shape into our Voronoi tiles, or we tend to look at these radial plots. And so we do see that this both shape index and dynamics, which I'm not showing here, but you can imagine RMS speed will show these consistently decreasing trends. And so to get back to a question that was asked earlier, probably you have leader cells that are starting to degrade the matrix, and then you have proliferative pressure that is pushing behind this. And so we always see this consistent trend towards jamming, if that makes sense.
All right. Thank you very much.
Thank you.
Great. We've got a few more, and thanks for everyone who's still sticking around to keep asking and answering or hearing these wonderful questions. So one more from Zhang Chen, wondering about your different spheroid states, solid, liquid, gas-like. Is there any difference in differentiation of these cells?
That's a good question. Not that we know, but we didn't directly test for that. So to the best of our ability, we have done some immunofluorescence on these. They seem to maintain their phenotype, but I was recently reading a work where collective invasion in MDMB31 spheroid is also associated to upregulation of E-cadherin. And so maybe the picture may be even a little more complex than obviously our simplistic study picture depicts it.
Thank you. Next question is from Anna Pasapera. Did you consider the role of MMPs that are highly expressed in mesenchymal MDA cells that play a big contribution in the invasive phenotype compared to MCF10, which are less invasive and more epithelial in nature?
Absolutely, that's a great question. We did not, primarily because this was already shown by others. Again, I've been referencing the work of the group of Peter Friedel, but also by George Yoshida in the past, and this was shown clearly. So we do see that, for instance, this type. I don't know if you can still see my my mouse. This invasive phenotype is primarily mediated by contractility, whereas this is primarily driven by MMPs. And so if you inhibit MMP activity, this will not, this collective will not invade in the same form, whereas this will not be affected. And so we did not directly tackle that question because we felt it was already addressed, and we wanted to sort of understand the structural signatures associated with different types of migration. But that nonetheless is a great question.
Thank you. Next question is actually from Suraj, which I think you can ask Suraj.
Yes, Suraj.
Oh, I mean, it was a very quick question. You might have already answered it. Great talk. So one was a clarification about, so there were these non-confluent sort of liminal spaces inside the spheroids. What's over there? Is it just fluid that the cells pump in, or something else? And the final question was, how much of the shape of the cells at the boundary of the spheroid, how much of that is correlated with the alignment of this ECM cage that forms in some cases and doesn't form in other cases?
Absolutely. So first of all, to address your first question, this was a slide that I removed just to address the fact that invasive cells, that is the MDMB31, form more liquid-like spheroids. That is, if we don't give them a little bit of matrigel, I believe this is 5% volume, 0.5% volume, sorry, they won't even form a solid spheroid. And so upon further analysis of the structure of these spheroids, we see that on multiphoton microscopy, they have a hollow core as opposed to the MCF10A spheroid. And this hollow core seems to be filled upon histological staining with matricellular proteins. So the very simplistic picture in my mind is that this cell type is has very low cell-cell adhesion, as was mentioned by George Yoshida, but they really like the matrix, and so they will compact around a matrigel matrix. And that's why the core is sort of hollow, is filled with matricellular proteins. Related to your second question, sorry, I forgot. What were you asking?
Well, it was to do with, because you had this variation of the cell shape with the difference between the core and the surface, and I want to know how much of the cell shape on the surface of the spheroids was correlated or related to how compact the ECM is in their vicinity and how aligned the ECM fibers are.
So I think what your question is referring to is, since we're estimating cell shapes this way, but again, as I mentioned before, we see this compact layer of collagen. Do we have tangential alignment also of the cells?
Yeah.
Right.
Yeah.
That's what you're hinting at.
Yeah.
Not by our measurement. So we don't seem to see a preferential alignment that is tangential, and that may be partially because of limitations of our method for cell segmentation, but we don't seem to see that preferential alignment there. Interestingly, a recent work by the group of Joseph Cass showed that in I think still in MCF10A spheroid, they saw more alignment that is tangential, but in slightly different conditions. So the model is similar, but not quite the same.
Okay, thanks.
You're welcome.
I think we have addressed all the questions, right?
Awesome.
Thank you so much, Suraj and Jacopo.
Thank you.
Great. All right, I'm gonna go ahead and
stop the
live stream. So the recording will be over, but before we do that, just want to say thank you to everyone for being here. Thank you to our